Hello and welcome to the inaugural lecture of our video series to understand rather than learn statistics and genetics. Today we will talk on the topic insights into biostatistics. Friends, the term statistics has different origins. It is said to be originated from a French word statistique, a German word statistic, Italian word statistica, and a Latin word status. These all words mean political state or a government. But what is the relationship of any political state or a government with statistics? It's because this branch of science was initially used exclusively for generating and analyzing data for government agencies, including birth rate, death rate, mortality rate, census measures, and others. What actually is statistics? It refers to the study of methods and procedures for collection of data, which can be done through different methods, randomly or non-randomly. Classification, which means grouping of data based on certain common characteristics. Tabulation for purposeful analysis in the next stage. Analysis of data using different statistical tests. And finally, interpretation of data. All steps, starting from collection, are important for final, meaningful, and reliable interpretation of data. Coming to biostatistics, it refers to the application of different statistical methods and principles to solve biological problems. These problems may be related to human biology, medicine, public health, agriculture, veterinary sciences, genetics, and other fields of life sciences. Biostatistics is also known as biometry, which means biological measures. Biometry consists of two words, bio and metry. Statistics can be divided into two main subcategories, that is, descriptive statistics and inferential statistics. Descriptive statistics mainly relates to summarizing the data so that it's comprehensive and more informative. It involves summarizing data using tables, graphs, charts, etc. However, it's not concerned with making inferences or drawing conclusions. On the other hand, in inferential statistics, generalizations are made and conclusions are drawn. Basically, in inferential statistics, inferences are made based on underlying hypotheses, statistical tests, and subsequent analysis. After basic definitions, we should know the basic terminology used in statistics. First is concept of population and sample. Population is well-defined group of individuals that are being studied or observations are being made on them. On the other hand, sample is a subgroup of population which is regarded as representative of entire population and on which the actual experiment is done. How sampling is done and what are the different methods of sampling we will discuss in later lecture videos. Next, we come to the concept of variable and constant. Variable is any quantity or character that varies from one individual or subject to other individual or subject. For example, human beings may vary in their age, weight, height, etc. So these all characters, age, weight, height, are called as variables. Constant is any quantity whose value is universal and does not change from one member of group to other. For example, seasons in a year, hours in a day, value of pi, etc. All these things do not change from one individual to other. In statistics, we mainly deal with variables. There are different types of variables, that is quantitative and qualitative variable. Quantitative variables are also known as numerical variables. These are measured on numerical scale. Quantitative variables are recorded using two scales of measurement, that is interval scale and ratio scale. Examples include weight, height, length, etc. Qualitative variables are also called as categorical variables. These are basically quality or attribute variables and cannot be expressed as numbers or in magnitude. For example, blood group, fragrance of a flower, beauty, allergic response in a person, etc. These variables are measured based on nominal and ordinal scales. What are these scales, how they differ, and what are their properties we will discuss in coming lecture videos. Quantitative variables can in turn be divided into discrete variables and continuous variables. Discrete variable can take only specific integer variables, that is 1, 2, 3, 4, etc. and so numerical values. No decimal values are possible for discrete variable. For example, number of children, number of doctors, number of voters in an election, number of electrons in an atom, etc. Continuous variables are those quantitative variables which take any value within a certain range. For example, weight of a newborn child, height of a person, lifespan of a person. There can be any value with decimals being possible for continuous quantitative variables. Coming to descriptive statistics. Two types of measures are included in descriptive statistics, that is, measures of central tendency and measures of dispersion. Measures of central tendency mainly stress on central values of the data series around which all other values spread. Measures of dispersion are also included under descriptive statistics. These give us an idea about the variability of data. Measures of central tendency mainly cover three kinds of average. Mathematical average, that includes arithmetic mean, geometric mean, and harmonic mean. Positional average includes median and mode. Median is the midpoint of a given data series, while mode is an entry which occurs the highest number of times in a data series. Partition average help in partitioning the data into certain sections or parts. Quartiles, values in dataset that represent 25% of a given population. Deciles are values that define 10% of a dataset. Quintiles are values that define 20% of a dataset, and percentiles define 1% of a dataset. The measures of dispersion can be distance measures or average deviation measures. Distance measures mainly include range and quartile deviation. Range is the simplest measure of dispersion, with it being calculated simply as difference between the highest and lowest estimates of a data series. Quartile deviation is also included in distance-based measures of dispersion. Average dispersion measures include mean deviation, standard deviation, and variance. Variance and standard deviation are two mainly used measures of dispersion, though they look similar in meaning but are different. This will be discussed in later videos. The other measures of descriptive statistics include measures of skewness, measures of kurtosis, and coefficient of variation. Normal distribution is symmetrical with its mean, median, and mode coinciding at a single point. The distribution can be positively or negatively skewed. Measures of kurtosis signify the peakedness of a distribution curve. If a peak is normally curved, the distribution is mesokurtic. The distributions with peaks greater than normal are called leptokurtic, while the distributions with lesser peak than normal are called platykurtic. Coefficient of variation relates the variance with mean in an interesting way. If we were to compare the weight of an elephant to that of a rat, the average weight of an elephant is around 4000 kgs, while that of rat is only 200 grams. Now, if there is a variance of 100 kgs in the weights of a sample of elephants, it will not matter much. However, if there is a variance of 100 gram in weight of a rat population, it makes a big difference. So, coefficient of variance is one such measure which provides us an idea of variance in relation to mean of a sample or a population. Inferential statistics refer to processes and methods in statistics that help in generating conclusions and inferences. It starts with making a plan of work and setting a hypothesis. This is followed by hypothesis testing and different significance tests. This gives us a test statistic which is compared to critical values, and set hypothesis is accepted or not accepted. Different assumptions are regularly made for arriving at meaningful results at the end of a statistical test. These assumptions are mainly related to the sampling process, underlying probability theory, underlying distribution, and other aspects. Based on these assumptions, a statistical test may be a parametric test or a non-parametric test. Parametric tests include z-test, t-test, ANOVA, and chi-square test, while the non-parametric tests include rank sum test, Spearman's correlation, Mann-Whitney's U-test, and chi-square test. Chi-square test is considered both a parametric and a non-parametric test. In the next few video lectures, we will try to cover more basics of statistics and clear some of the common misconceptions involved in it. Friends, if you like the video, kindly share it among your friends and help us share the knowledge of statistics and genetics in a better way. If you have any questions, you can write to us. Suggestions and feedback are also welcome.